Perfect. And, and we have two speakers in today. So we're excited to have our two speakers, Dr. Paxton and Mr. Chris Cravante, both whom are medical technicians and also teach in the Department of Biological Sciences here at Rowan. So the format for our seminar is going to be this. We're going to have each speaker present a description of their field and their own personal career trajectory for about 20 minutes. After both speakers have presented, we'll have about 15 minutes of question and answer. So we do encourage you to put your questions in the chat. So if you check your chat feature for those that are on Zoom in the classroom, obviously this is going to be a little bit different. But if you check for those on Zoom, again, if you check your chat feature, you'll see that you have, or you should have, either 2 or 3 options. And please make sure that you send your questions to Dr. Krummenacher, who will be organizing the questions and then asking the questions to the speakers once we're done our presentation. So again, please make sure you put your questions in the chat. If you're doing, or if you're submitting questions, if you're submitting questions as part of your course, so it's a requirement from your instructor, then please make sure that you indicate that instructor's name in the chat. So this way we can relay to them that you participated in the presentation. All right. So again, I'll give some reminders during the presentation as far as when there's 5 minutes remaining, 1 minute remaining. So this way we can stay on, keep kind of time management. I know that, our instructors have other courses to teach and the students also have other classes. So we just want to make sure that we don't go too far or we don't go over time. So I'm pleased to introduce our first speaker. That's Dr. Paxton. She is a 3/4 time faculty in our bio department, a certified medical laboratory technologist, but also has years and years of experience in the field. So I'm going to let Dr. Paxton take over the presentation and feel free to share this with us. Thank you.
Hi, everyone. I'm really glad to be here today. So we're going to talk about careers as a laboratory scientist. And as most of you know, when you start college, you don't necessarily know where you want to go. You decide to major maybe in biology or chemistry, and you get to the end of your senior year and you go, okay, what am I going to do now? And literally that's what happened to me. English is my second language, so I was still learning English when I graduated from college, and my goal was originally to go into medicine. But because my English skills were not so good yet, it took a while to get the English under control. I was not able to get into medical school. And this was 50 years ago. So remember, I'm old, okay? So I decided that I would get my master's and then I would go into being a med tech because I could get a job. Now, if I could get the PowerPoints to move, this would be good. Why is it not moving? My slides are not moving. Janine, can you control the slides?
No, I cannot control the slides. Did you try the page down?
All right.
Oh, there we go. There we go.
Okay, there we go. So I went to a school in Toledo. It was part of a hospital system. And if I could get rid of what's on the screen here, I decided to go through a medical technology program so I could learn to be a lab rat, right? And we hear a lot about nursing programs. We do not hear a lot about medical technology or medical laboratory scientist programs. Most of the programs are through the ASCP, American Society of Clinical Pathology. So the ASCP unites more than 100,000 anatomic and clinical pathologists, residents and fellows, medical laboratory professionals, and students to advance laboratory medicine to better improve patient care through knowledge, collaboration, and a global effort. It's just not moving. So what is medical laboratory science? Okay, it's a branch of healthcare. It's a laboratory tests that are used to diagnose and treat diseases. These tests are all certified and validated by the FDA. They're used to monitor health. And the medical laboratory scientist is a trained professional in the field. Usually this person has achieved a bachelor's degree. So what do we do? Okay, well, we work in clinical laboratories. So what's a clinical laboratory? It can be a hospital laboratory, a physician's office. It can be a research and public health lab. It can be a variety of departments in, say, at Jefferson or Rowan or wherever you might go to work. And you can also get into state health departments, the WHO, the CDC. You can do water quality. You can go into the food industry, a whole variety of career paths once you get your certification. So the fields, as it was presented to me way back 50 years ago, there were 7, at that time, there were only about 5 fields. We had clinical chemistry, medical microbiology, hematology, clinical immunology, immunohematology. But since I was certified, we now have molecular diagnostics and flow cytometry. So obviously in clinical chemistry, you're going to look at bodily functions that have to do with enzymes. Medical microbiology, we're going to be looking for viruses and microbes causing issues. But one thing that's very, very interesting today is medical microbiology is turning more and more into molecular diagnostics. Many of the diseases that we usually do cultures for are now diagnosed with PCR or other rapid techniques. Hematology, all of you probably have had a total white blood cell count done, red cell count done. It's looking for abnormalities, looking for leukemias, lymphomas, anything of that sort. Clinical immunology, again, response to disease. This can be serum-based, it can be cell-based, and this flows very nicely into flow cytometry. In fact, I spent 15 years of my life doing flow cytometry for the HIV epidemic and doing all the work for GW for leukemias and lymphomas. So many of these fields sort of meld together, but as you go through your training, you have to become proficient in all 7 fields before you can take your certification exam. So the medical laboratory scientist has many duties. Okay, you're going to do advanced testing on specimens. You're going to do molecular or microbiology testing. You're going to confirm blood types and determining transfusion eligibility based on antibodies and the presence of antibodies that could cause a reaction with a blood transfusion. You're going to learn how to use computer operated technologies, which we have many of now. And we're going to really have to explain to our physicians or to our nurses what the testing is and what the abnormalities are. So the medical laboratory scientist has a great role in really teaching others in the hospital system. Okay, so what are the paths? The requirements to get certified is, I'm sorry about the screen is hiding that the first, I can't get it to go up. So the primary path is to get a baccalaureate degree from a certified, a regionally accredited college or university. And then you have to go through, and usually that's a 4-year degree. Some programs have allowed 3+1, but most are doing 4+1. So then you go through the 4 years, and then you go through a NACLS accredited medical laboratory scientist program, and you have to do this within 5 years before taking your exam. The prerequisite courses are 16 semester hours, 24 quarter hours in biology, including 1 semester in micro, 16 semesters in chemistry, 1 semester of organic, 1 semester or biochemistry, organic or biochemistry. And this can be done within your baccalaureate program or outside of it. I can really tell you that if you don't have those courses, you're not going to make it through the program, because the NACLS 1-year clinical program is quite difficult. So it only wants to go backwards today. Okay, so one of the things that's really important to know is that once you get through your 1-year internship in the NACLS program, You can also get certifications in blood banking, chemistry, histology, microbiology, cytotechnology, hematology, cytometry, and molecular biology. You can also get certifications in diploma in laboratory management, which is a DLM ASCP, and that's usually they require a PhD for that. You can also do a pathologist assistant program, as known as PA ASCP. So the routes to get to these are several. They require either the MLS, a medical laboratory scientist certification, plus experience, or a master's in the field plus experience. Okay, so you might want to ask, are there a lot of jobs out there? Okay, the top medical laboratory scientist jobs. One of the things that really is needed to do this job is a high degree of self-motivation. You have to realize that you are going to have to continue learning and learning and learning as the years go by, because as you know, the technology keeps changing. So you can be a medical laboratory scientist, you can be a clinical laboratory scientist, a medical technologist, a clinical laboratory technologist. They all have slightly different definitions. A clinical microbiologist, a laboratory supervisor, a supervisory technologist, a forensic scientist, and you can also be a laboratory director. You also have the opportunity to get a variety of state laboratory certifications. New York State is one of the big states that requires additional training. Once you get those licenses, you can basically work anywhere. So what are your work locations, okay? There are multiple settings. While most MLS graduates seek employment in hospital laboratories, other options are available. As I mentioned before, forensics, biotechnology programs, toxicology, medical research, healthcare administration, and pharmaceutical development. Once you have those credentials, it basically gives you that license to go work almost anywhere. So the profession as a whole has grown exponentially, 16% by 2024. This is faster than the average growth. And this is, I'm sure that COVID has added to that burden. The median salary in 2015 was about $50,000, but it's expected that for 2021, the median starting salary is between $65,000 and $84,000. Okay, there are several societies: American Society for Clinical Laboratory Science, the American Society for Clinical Pathology. These are the societies that you would belong to if you were to go this career path. Okay, so you might wonder, okay, how did I end up being a faculty member at Rowan? Okay, so I graduated from college in 3 years, and basically I didn't know what to do with myself, and I couldn't get a job. So I got my master's, I got an MS in microbiology. Then I went and received my ASCP certification in a hospital setting. So then I was able to get my certification, and I moved to Colorado Springs and worked as a med tech, which is now known as an MLS, medical laboratory scientist, in a microbiology hospital in Colorado Springs. Well, those of you that know me know that I get bored pretty quickly. So I decided, after being a med tech for a while, that I wanted to do something different. So I was hired as a product manager for an immunological products company, and I moved to Texas. In that particular job, I learned a lot about immunology. I did a lot of autoimmune disease testing and actually developed tests that are out on the market now for anti-nuclear antibody testing. And I also developed the first IFA test for toxoplasmosis that you use for pregnant women to make sure that they've been exposed prior to being exposed to cats if they're going to be in their first trimester. So out of Texas, I was called by someone in Baltimore, and they asked me if I would be willing to come work in Baltimore and to set up an immunology laboratory, which I forgot to put this on the slide. But at that time, I had started another company that was an immunological products company. And there we made FDA cleared tests for all kinds of things, such as dengue, leptospira, malaria, yellow fever, Rocky Mountain spotted fever, rickettsia typhi. Anything that was immunology based, we attempted to do. And I was given over $1.5 million grants by the DoD, the Department of Defense, to develop these tests for the US Army and Armed Forces. So the FDA Immunological Products Company, it took time. So I hired about 45 people and we developed these kits. But at the same time, I was working as a laboratory director for 15 years in this immunology lab, which was a commercial lab, which is now part of Quest. Again, my skills as a laboratory person were really helpful because it allowed me to be able to teach and it allowed me to be able to actually do the assays myself. So I was laboratory director for 15 years in an immunology and flow cytometry commercial lab. I did all the work for GW for all their leukemias and lymphomas, did that for almost 10 years. And then lo and behold, I got cancer. So I was worried about my staff and I was worried about the fact that I had this company and if I died from cancer, I didn't want them to be stuck without a job. So I sold my company to an Australian company and those products are still being manufactured. But then I got bored because treatment for cancer is not a short thing. So I finished my PhD and then developed did some courses in epidemiology and became certified in epidemiology and became certified called SIC, certified in infection prevention and epidemiology. And I started working in a hospital. The key thing that I'm trying to point out here is that every job I had, the whole basis was laboratory medicine and laboratory science. So there was no wasting of time. Everything built on each other. So I worked for hospitals as an infection preventionist epidemiologist for 14 years. And at the same time started teaching at Rowan, South Jersey, and at Rowan, Glassboro. So here I am today as a professor in the biological sciences. So basically that ends my presentation and I would welcome any questions.
Thank you very much, Dr. Paxton.
Did I do it on time? Yeah.
Absolutely, you're fine.
Okay, good.
Thank you. Thank you again. Please make sure that if you have questions, that you send them through the chat to Dr. Krimenacher, and he will organize our questions and then ask them to the presenters once Mr. Gravante is done his presentation. I know in the classroom, you're not going to be able to submit your questions in the chat, and that's okay. So just if you could hold your questions to, again, once Mr. Gravante is finished, then we can open up time for in-class questions as well as Zoom questions. All right, so thank you again, Dr. Paxton. Our next speaker is Mr. Chris Gravante. Mr. Gravante is a certified HLA technologist at Temple University Hospital, and also an adjunct instructor at Rowan. So thank you, Mr. Gravante, for being here with us today, and I'll let you get started on your presentation.
Sorry, I just can't share my screen until Dr. Paxton gets rid of.
Yeah, I'm trying to unshare here.
Okay.
Yeah.
Let's see, new share, it says new share, stop sharing, where's stop sharing? Stop share, there we go. Sorry.
There we go. That's okay, that's good, that's good. All right, so I'll share mine now. Okay.
Thank you.
And then, let's see, I should be able to, sorry, it's the screen is blocking it, oh my gosh.
Can you go down to the bottom there?
There we go. Play from the start. Here we go. All right, awesome. So, all right, we're good. We're good. We're good. So my name is Chris Gravanti. I'm an adjunct here at Rowan University, and everyone can hear me okay, right? Make sure we're good. Okay, awesome. So I'm actually going to talk a little bit about my career path first, and then I'll jump into exactly what I do. So my career path is kind of went a little bit of different directions. I actually started at Rowan Community College, which when I started it was Burlington County College. And then I transferred over to Rutgers University. At Rutgers University, I didn't know what I wanted to do. I was in many different directions. I had no idea what I wanted to do. I thought pharmacy. I thought a whole bunch of different things. So I just started taking as many science classes as I possibly could take because I knew science was where I wanted to go. I ended up getting a BA in general science, which just means I've taken plenty of chemistry classes, physics classes, and biology classes. I got a BS in biology because I had the most in biology. I ended up doing a lot of research in biology as well. We used to do a lot of leech research back in the day. I used to sequence leeches with PCR. I used to do it by hand. If anyone's ever done PCR by hand, it's not a fun time. I also did a lot of histology work. Used to make slides and stain them, which we used to use a microtome. If anyone knows what a microtome is, it is also not fun as well. So, but and then I ended up actually getting a certificate in secondary education because I thought maybe I want to teach biology. So, and that's where I ended up teaching a half a year at Cherokee High School. So at Cherokee High School, and I found out very quickly that I did not like the politics in high school, in a high school setting. So I decided to, I got a phone call from actually a job that I used to work at, and it was called Labs Inc. at the time. And that's a lab that's located inside Gift of Life. If anyone's familiar with Gift of Life, they're one of the big transplant organ operation systems in the Delaware Valley. So if you know anyone who's got an organ transplant or anything of that nature in this area, Gift of Life probably played a part in it. During that time, I decided I wanted to elevate my education. So I actually applied at Johns Hopkins University and got accepted into there, and joined their I joined their immunology program and then specialized in biotechnology there because I was actually really interested in a whole bunch of things that I was doing in that lab at Gift of Life. It was pretty much a general lab. So I'll just continue on and then I'll actually give you my recommendation at the end. So after 2 years, I traveled a lot around the United States. I've actually been to a lot of different transplant labs within New England. So that would be in Boston or Whaler, well, something like that. And then I've been to Denver and Colorado. That was our home base out there. I've been to Atlanta to work with helping their patients. And actually my favorite city of all was Minnesota, which I never would have thought that, but I remember being in the winter storm of -40, and that's the cold that can go through 2 jackets, and it was very cold. But it's very interesting to watch a whole bunch of different types of labs and how they function. And it was nice that I was able to do that and go around. And that was all because I actually interned at this place, Labs Inc., my junior year. And so I would highly recommend if you could get an internship in a lab, paid or unpaid, just to get your foot in the door. Because after I graduated, that's why they called me and said, "Hey, do you want a job?" But I was there for about 2 years, and then I got my certification in HLA because that was one of the things we did there, and that was actually my favorite part of the job. So I got certified, and then I applied for Temple University Hospital, which is where I still am today. I also knew the manager over at Einstein, so she had called me and asked if I could help them out because Einstein is closing and merging with Jefferson. I think it's officially now merged with Jefferson, so who knows if I'll still be there or not. We don't know. Not a big deal because I'm also an adjunct here at Rowan University now, which is where I am today, along with Temple and still technically at Einstein. One of my big recommendations for people who are interested in the medical technology role would be find a lab that has many different avenues. So when I was at VRL Eurofins, Labs Inc., Gift of Life, whatever you want to call it, there was a very generalist lab. So we did infectious diseases, we ran blood typing, we would run HLA and do like microbiology work as well. So from there, you as a generalist, you might not make that much money because you're a generalist and you're not specializing in anything specific. But there I was able to find my passion in which area that I like the most. Um, so from there, I realized I liked HLA, so I went and pursued that direction. And then once you specialize in any specific position, the money comes in, and then you start actually making the money that you want to make, that you feel like you're entitled to because of all that schooling. But the big thing is the big takeaway for me was it wasn't really the MS in biotechnology that got me the big pay raise. It was actually the fact that I was certified with ASHI, which is American Society of Histocompatibility and Immunology. It was the fact that I was certified in there, which got me the job at Temple, not really the MS in biotechnology. That helped me get the adjunct role though at Rowan. That was it. So I just want to talk a little bit about transplant information in general. So the transplant labs are very fast-paced work environment, and I'm going to focus on transplants because that's what I do at work. Is I focus on transplants. So it's a fast-paced work environment, and because the shelf life of these organs are very short-lived. The patients, a lot of these patients are on life support, and you don't have a big decision, you don't have a big window of time to figure out where they're going to go, because you have a whole bunch of different hospitals in the Delaware Valley that are looking to get their organs for their patients as well. So what happens is a patient gets diagnosed, a donor gets diagnosed, and they're usually pronounced brain dead at that point. And this is where it gets kind of morbid. But the way I look at it is it's one family's worst day, but it's the next family's best day. It's the best case scenario for that other family. And a lot of times the family discussion will happen, and that will be when if it's accepted or denied that the family is going to donate that organ into, go into organ transplantation. So when I was at Gift of Life, we would do the evaluation of that donor, which is why we would do a bunch of serological typings, infectious diseases. We would give out the HLA and the blood bank and the blood typing as well. Now over at Temple, I do what is the matching part, which I take that information that was given in this medical evaluation, and I take it and compare it to all the patients that we have on our list. Now the list actually exists, and it is very long, and there's plenty of patients on it that have an opportunity to get transplanted. But we give them statuses. So in a whole database of different statuses, there's patients that are status 0, which means they need an organ as fast as possible. If they can get the organ, then if they're a match with that, they're going to get it because they take priority. That could mean that they only have a couple weeks left, and this organ is going to be a life-changing opportunity and things of those natures. And then as you go down the statuses, they're less and less and less priority. Status 7 would just mean that you're on the list, but you're not actively looking for a transplant, but you're on there and you're following all the protocols. Status 8 would mean that you're unfortunately removed from the list, and there could be many reasons why. Maybe you're not doing everything you're supposed to be doing. Patient has passed away. That would also qualify you to be removed from the list. So over here, you can see that the job is, as an HLA tech, it's flourishing still. So we look at 2011, we see that we had barely any lung transplants. We had 0. We ended up getting a really great doctor, a really great surgeon over at Temple, and our numbers have skyrocketed since. And if you even look at 2020, the numbers dipped a little bit, but we were pretty much shut down for 3 months at that time. I was actually doing COVID testing during this time. I wasn't even doing HLA work because they just needed everybody and anybody who knew how to run any form of PCR to just come in and just do testing for COVID. So there's always opportunities there as well. But even with those numbers dipping just a little bit, we could see that the numbers are still pretty much on the rise. So what is HLA? So if you've taken any immunology courses, you may have mentioned it before. It might be briefly mentioned in some other courses, like a paragraph or two. But essentially what HLA is it defines who you are. So it's located on chromosome 6. So you have 23 chromosomes from your mom, 23 chromosomes from your dad. So you get half of your HLA markers from your mom and half of your HLA markers from your dad. In transplant industry, we look more for class 1 and class 2. Class 3 has no effect on transplantation at all. And one of the big things that I like to see is if you have a family member who needs an organ, it's usually your siblings would be your best opportunity because you get half from your mom, half from your dad. Odds of you being most similar would be with your siblings. So continue on here. So what is the role of an HLA technologist? So an HLA technologist has many different aspects and many different roles, but to summarize it, is our role is to find out what a patient's HLA typing is. So our patient's HLA typing, and we're going to compare them to donors' HLA typings. So it's almost like a crossword puzzle, essentially. And in a perfect world, in a perfect situation, we would want to match them up, what we call an 8x8 match. And that would mean that I want my A, B, C, DP alpha, DP beta, DQ, DP, all to be matching together in a perfect world. This is obviously a very challenging task to do, and we're not always going to have that perfect match. Actually, most of the time, we do not have that perfect match. But we use, at our lab, we use what's called next-gen sequencing. So next-gen sequencing is, it's very specific sequencing, and it actually is, it's really intense that you can sequence a whole genome literally in about 20 hours. It's insane. This is my HLA typing right here, which I think is cool because I get my own. And then you can actually take your own HLA typing and you can actually put it into what's called haplostats, and they'll actually determine where you come from, which is always nice. So I always look at mine. My mom's side has her dad is Native American, and you can see the NAM part actually elevates over there too. So I actually think that's pretty cool that it actually catches that side. Obviously, I'm Caucasian, so that's obviously there. And you can see like some Asian specific islander in there as well, and different types of things. So it's definitely going to be linked back. We definitely see commonalities where people come from and a whole different bunch of things like that. And I threw in the central dogma of molecular biology because I want you guys to realize that just because I have a DNA part, remember the DNA will make the RNA, which will make proteins. So DNA to RNA to proteins. So if I have an A1 allele, which is what I am, I'm A101 and A3101. These will also make those protein receptors. So my protein receptor would be A1 and my other receptor would be A2. They're co-dominant, so the one's not going to overexpress the other one. So on my outside cell markers, I should have an A1 and an A31 on the outside of it. So whenever I can interchange these in between. I'm talking about alleles, or I could talk about antigens and antibodies. So that brings me to the second point, which is to find out our patient's antibody profile and then compare them to our donor's HLA typing. So if I'm not going to get the perfect match, I want to make sure that the patient does not have any antibodies to the donor's HLA typing. Because if that happens, I might have an instant rejection. And that's pretty much what the main role of an HLA technologist is. So how do we do this? So we're going to monitor patients every month, essentially. So as your role as an HLA technologist is you're going to get very familiar with patients who are on the list. I know a lot of them by name. I feel like I know them. Never see them though. So that's the beauty of it. You don't actually have to see the patients, which I like that part. I'm kind of hidden in the background. So we use a lot of, Dr. Paxton mentioned that she worked with flow cytometry. I also work with flow cytometry. And I think what you'll notice is that a lot of labs, flow cytometry is the way, it's definitely the method that goes a lot now. Flow cytometry, PCR, all types of different amplifications is the way the lab is going nowadays. So for DNA typing, so typing is how we get their molecular typing, which would be the DNA side. We use something called NGS at our lab, which is next-gen sequencing. When I was over at Gift of Life, we used something called real-time PCR. It was a lower resolution typing, which is like you get pretty much just get a generalization. This is probably what it is. That only took about an hour to do it, and it was very fast. It was very quick. We also use specific sequence primers. We also use oligonucleotides as well, SSO. And those are all done through PCR methods. SSO is done through flow cytometry as well. SSP uses gels. So if you've ever ran a gel before and the bands go up, people still use that method out there. The antibody testing is done through flow cytometry, and you get almost this chart results right here. Anything in red would be a good indication that it's probably not a good idea to go through a transplant. I believe I am a B51. So if I was on opportunity to transplant with this patient, my organs would not be a good decision to give this to this patient, as you can tell with the B51 being in complete red down here. Can you guys see my mouse as I move, or not really? Yes, okay, awesome. So there's the B51 and you can see it elevated here. And that's just a quick summary of what our role is here, but we also have what's called our final testing, and that is called cross-matching. And this is our big test that we do at the end to kind of summarize everything to make sure it works. There's two different types of methods that people still use. That is CDC cross-matching, and it's also flow or fast cross-matching. My job, we do flow cross-matching. I think a lot of the directions of the lab are going flow cross-matching. I don't think anybody really does CDC anymore, but I have seen it in Einstein. So I'm just going to explain what cross-matching is, because I love images. If anyone, any of my students are here, they know I like breaking down things and going into detail. So I love looking at the way mechanisms work, and I love breaking them down. So I'm going to explain how flow cytometry works. So whenever you guys go in there and go get a job, and you can be like, I know how to do flow cytometry. I know how it works. So cross-matching is very easy, actually. It's the idea behind it is actually really simple. We're going to take our patient's serum, the patient that we want to give the organ to, and we're going to just put patient's serum into a well. So if patients have their serum in there, what is located in their serum is they have antibodies. So if we see recipient serum here, we can see the antibodies that would be located inside that serum. And then we're going to add the donor's cells into there. When we add donor cells in there, if any patients have any antibodies to those donor cells, they should connect, almost like a puzzle piece. They should connect. And if they connect, we can actually add something. We use FITC as our color reagent, would brighten it up and brighten up your antibodies so they show. The only other thing we have to do is locate, we use T and B cells. The only thing we need to do is make sure we mark our T and our B cells. If we mark our T and B cells, then we'll know if it's T cell positive or if it's B cell positive, which will tell us if it's class 1 or class 2 HLA markers, which is a good indication of transplant can go on or not. We do that with what's called CD markers. So if you're familiar with your cells and you know all the CD markers off the top of your head, you'll know that CD3 is a pretty good indication that you have a T cell. And if you use CD19 or CD20, it's a pretty good indication that you have a B cell. So we'll throw that in there too. And you'll get your results look almost kind of like this. Anything to the left would mean that no color has been picked up, which means you had no antibodies bound to that cell. If it's shifted slightly, then that means you have slight positive and a slight reactivity. If it's fully shifted, you should probably avoid transplant at all costs because it's not a good opportunity. That would mean that it's very strongly positive and you should expect rejection immediately. We did a similar idea with the beads that you saw in the previous slide with flow cytometry over here. Same idea with the beads. You add the patient's serum. Instead of adding cells, you add beads. Antibodies bind to those specific beads. Those beads can contain all of A2, AB44, all different types of markers, and they also run through a flow cytometer as well. So essentially, we're pretty much that final say. So after we've done a whole bunch of things, a bunch of workups with that, with the OPO, which was like gift of life, once we've done all that workup with them and everything is passed, we're the final say before the patient goes into transplant. And Dr. Paxton had mentioned that, you have to talk to these nurses and you have to talk to these doctors and you have to explain it. A lot of these doctors, they just want to know if it's good to go or is it's not good to go. So remember, they're going to take your word for it because you are the expert in that topic. They're not the expert in the topic. They actually don't really want to learn too much about it. They just want to know, is it good or is it not? And I think that's anywhere in the lab. They just want to know if it's ready to go. On that note, that's pretty much what I have for you guys today. If you want more reading to do, I did just have a publication out with Temple. We discovered a new allele that did not have any start sequence, so no ATG to start it off. It was a synapse mutation over, I think the T switched to a C, if I remember correctly. So, and then obviously we'll take any questions as well, and anything of that nature. So I really wish you guys the best of luck though, because it's a great career avenue to go into, and I really think you guys will like it. It's very nice. So that's all I have for today though.
Well, thank you very much, Mr. Gravante, and thank you again, Dr. Paxton. I agree. This is a very exciting field, and I think that, you know, I appreciate you bringing this information to our students. Excellent presentations. Thank you very much again. So at this point, we're going to start our time. We're going to start our time for question and answer. Again, please, if you're on Zoom, please post your questions to the chat and send them directly to Dr. Krimenacher, and he will share those questions with our speaker. And then for students in the classroom, if you want to ask your questions directly to, to Dr. Paxton or to Mr. Gravante, then we can do that. But let's start, I guess we'll start with the Zoom questions first, and then we'll move to the classroom. This way we don't have everyone trying to kind of ask questions at once. So Dr. Krimenacher, I'll turn this over to you.
Thank you. We had a couple of questions already in. The first one I want to ask Mr. Gravante about his work, and somebody asked if it's possible to work at home since you essentially deal with patient data on a computer. And the second question on this is, how long does it take to analyze matching pairs of a donor and recipient?
Sure, that's a good question. We've been trying to get some work from home opportunity as well. You always need someone to be in the lab to run the assays. They don't, no one's going to ship them to your house so you can run them there. You do have to run them in the lab, but there is a lot of opportunity for analyzing. So analyzing could easily be done at home, and I wouldn't be surprised if that direction started to come with some working from home opportunities. I can see, I have, my manager works from home a lot. So if you want to be like a supervisor or anything along those natures, I mean, a lot of managing is just going through the data and going through making sure everything is done correctly. And that opportunity for working from home is definitely there. But the job also inclines on call. So you do have to go in and if there's a death over the weekend, you do have to go in over the weekend, and, you know, the opportunity calls cross match needs to be done. And to analyze a patient, it depends on the patient. Some patients have zero antibodies to any HLA determinants. They've never been exposed to any HLA determinants. They've never been transfused. The vaccines, no interactions with them. And they could take, really fast to analyze if they're negative. If they're positives, there's epitopes that I got to look for and things of those natures that can cluster up, and they could take to an hour, to analyze. A lot of the NGS software is much better now, so it used to take a long time to analyze before, but not as much anymore. I'd say about an hour max to do proper analyzing.
All right. Well, thank you. That was interesting details about this matching, which is absolutely amazing the way it's done and how fast it is and how important it is to save lives. There's another question that I want to ask to Dr. Paxton first, and then Mr. Gravante can probably chime in. And the question is essentially, how different is it to be a medical technologist versus other biotechnologies in research, for instance? So are they different practices, priorities, and the person asking the question also mentioned budget and ethics. So if you can compare essentially medical technologies with other biotechnologies.
I think, Claude, that it's a good question. Being a medical laboratory scientist implies that you have the training in the clinical sciences, the clinical chemistry, clinical hematology, immunology, et cetera. If you go into say you want to go into biotechnology and you want to make recombinant proteins, you don't necessarily need the certification. But nothing about the medical laboratory scientist avenue keeps you from being a biotechnologist in a biotech field. I would say to you that if you have those credentials, your flexibility as an individual is going to be much greater, so that in 1 minute you might be making a recombinant protein to dengue virus, or you might be making a recombinant protein to a cancer cell that you might want to make a vaccine out of it. So having the clinical background really supports your ability to do your job better.
Yes, I think, yeah, I agree with that. It opens more doors, I think, if you have the ability to do medical stuff.
Mhm.
And
Yeah.
a question to both of you, and probably Dr. Paxton can start. Can you elaborate on how certification is done? Do you have to take courses? Do you have exams? Do you have practicals? Is it something that you do online, or do you have to?
No, it has to be done. Certification, remember, it's a 4-year baccalaureate in an approved university, plus a 1-year clinical rotation through each of the laboratory departments. I mentioned 8 different departments. After you have finished your clinical in an approved program, you take a certification exam. And I do a lot of tutoring for this now, and the fail rate is about 75%. Okay, so this is a very difficult exam. You have to get a maximum of 400 for each field. So for blood banking, hematology, chemistry, microbiology, molecular, and flow cytometry, you have to pass every segment in order to get certified. So we find that the students that have just graduated, have taken their clinical rotation, are in a much better position to take their certification exams immediately after school, rather than waiting a year or 2. Because unfortunately, it's like anything else. If you don't practice it, you're not going to be able to remember it.
Yes, thank you. Mr. Gravante, you want to add something about your experience with these exams? And
Yeah, sure. To touch back on research-based as well versus clinical-based too. Research jobs, I've noticed, are a lot more laid back, a lot more relaxing than a clinical lab job. But the big thing, and I'm sure everyone wants to hear this, is that the pay is also much less. So if you want to make more money, I would highly recommend going into a clinical field. The money is more there than a research-based field. Yeah, the exams are tough. So I would highly recommend, as just like Dr. Paxton said, immediately afterwards, definitely take it. Don't lollygag it. Definitely take it right away.
So many of my students that I tutor, I may tutor them for 3-4 months, going back over each of the disciplines and doing the lab CE questions and going over the theory. And usually they end up taking it at least 3 times before they pass it.
yeah, not an easy thing to do. Then there's another question about, if there are any physical requirements or limitations to do the jobs that you are doing, whether it's standing a lot, sitting a lot, carrying heavy stuff, or if you have, limitations like being colorblind or anything that you may think may affect your job.
Chris, I'll take it first. I would say that it depends on the location of your work. If you're working for a commercial laboratory, if you're running a chemistry analyzer day in and day out, you may be standing a lot. One of the reasons that people don't go into the field sometimes is because they feel that everything is too automated and they have no control. So again, it's going to really depend as to the situation that you're working in. Usually there's not a 50-pound requirement or anything like that. You know, you might have to handle a box of saline or so on and so forth, but no, there's color blindness, may or may not be a problem. It would depend on the type of color blindness that you have. So generally, I know that in our flow cytometry lab, I had 10 flow cytometers and I had 2 hearing impaired individuals. And we basically had some concern about safety for those individuals. So we managed those individuals with lights that would go on if there was any kind of an emergency. So, you know, hearing loss is probably not a problem.
Thank you. Mr. Gravante, you want to add something about?
Sure. Speaking of hearing loss and color blindness, I did have to take a colorblind test to start the job. They do give you the cards with the numbers in them. But standing wise, I'm noticing a lot of the technology going to where it might be easier to run the assay and maybe it's much faster, but the actual data analysis portion is actually longer because a lot of the technology is giving you a lot more data than what it used to give you. So what you find is that the test might take you quicker, but the analyzing part takes you a little bit longer. So if you're worried about standing, of course, there's definitely lab jobs where you're standing all the time. I see people in microbiology running around like crazy all the time over there. But my job, per se, yeah, there's some standing parts, but there's also some sitting parts. So I guess it depends what field you go into.
Since you mentioned data analysis, is there any computer skills that are required or that you develop when you do this job?
So, I mean, I would say knowing Microsoft Excel is definitely a good feature, but not necessary. You get good training in any LIS system. They're all different. I've learned probably 6 of them now, and I've been 2 labs, so there's plenty of different ones. They change all the time. I've worked with LIS systems that are as basic as MS-DOS, and then I've worked with some that are as intense as you know, they knew exactly when we got the sample, when it was put in the fridge, who received it, and what time. Everything was time stamped. So I've worked from the most basic ones to the most intense ones, and I think just repetition, learning how to use them. You know, I think everyone probably in these classrooms are probably good with technology at this point. So I don't think there should be any issue there.
I do want to comment on that, Claude, because if you're working in a hospital-based system, you cannot use the excuse that you're not computer literate. You have to be computer literate to work in the laboratory.
Yeah.
The laboratory information systems have to coordinate with the EHRs, the electronic health records, that the nurses and the physicians are using, and all the orders and all of the assignment for tests are coming through the EHRs. So you have to be able to adapt to learning the different software. Also, flow cytometry certainly has a lot of software agility that has to be done in order to understand how to gate your population. So it's good if you have an aptitude for computers and gating and graphics for these jobs.
Okay, yeah, thank you. So somebody asked whether, and I think Dr. Gravante mentioned that you've been involved in the COVID-19 testing.
Mm-hmm.
Can you mention if you had to learn something specific for that, or how easy it was to implement?
Oh, yeah. So for COVID-19 testing, they've always did coronavirus testing before. So, but we needed to get new equipment for the COVID-19. And that process came very fast. So we started off with a very long method that was validated first, and it took a long time. It took us, we could only do like 48 patients in like 4 hours. So we needed all hands on deck. It was a reverse transcriptase PCR. And then it turned out that after a year, it was as easy as opening up a tube and putting it on an instrument, and you got your results back in a couple hours. So the technology just gets better and better every time. It's just a matter of when it gets validated and how you can use it fast. But our first method was very long and tedious. It was a long time, but it gets better.
Thank you. Just back to the computer skills. You recommend Microsoft Office over Google Docs and Google whatever spreadsheet and systems?
I don't know if the coding is similar in Google, but I mean, I'm talking loose skills in Excel. You don't have to make your own daily charts or anything like that. Just loose skills. Some of the equations are come in handy, but I think you can pick it up pretty fast, and it's not that hard.
All right, thank you. I don't have any other questions on the chat, but please continue posting them. And I want to ask Dr. Paxton if she has questions from the floor.
Referring to back to the technology that you need. Do you guys use a jump system? Because that's something that we have to learn a lot in biochemistry.
What kind of system?
Jump.
GMP.
Jump system, yeah.
It's like a statistical analysis software.
Yes, they literally kill us.
The question is being asked, okay, what's the name of the software?
Jump.
Whether or not we have to use a lot of jump software? Is that correct? I don't know the answer to that, to be honest. I don't think so. What about you? The question is, because in biometry, you have to learn the jump software. The question is whether in our fields, do we have to know that, Chris? I'm not aware of that.
So the softwares that I use right now in the hospital are you're going to use Epic software, which is what the nurses use. There's a software called Soft, which is what the lab uses. I use a specific software called Histotrack, which is what HLA technologists use. There's also, so I use 3 softwares right now. Jump is not one of them, but so.
They're all shaking their heads saying, why are we learning this?
Might help you learn other things. When I was learning coding, I had to learn MATLAB, and you never use MATLAB in your life, so.
Any other questions? Go ahead, take your mask off so we can hear you.
I'll just speak up. So for students like us that are in maybe like a biology major program, what would be the best path for us to pursue if we wanted to become medical technology?
Okay, so the question was, what is the best, if somebody is a biology major, what is the best path for them to become a medical laboratory scientist? Okay, and so here again, it's a baccalaureate degree, right, in biology or chemistry, as long as you have biochemistry, and you have microbiology or organic, okay, either pre-graduate or postgraduate, okay, and then getting accepted into a clinical program.
So would it be better to do like an associate's clinical program or get another bachelor's degree? I know there are some like certification programs that aren't degree-based.
No, you would have to get a 4-year BS degree, okay, baccalaureate, and then you have to take your 1-year clinical, and that clinical is both theory and practical, okay, and then you're going to have courses during that time that you will have to pass, and then you take your certification exam. Okay, now you used to be able to grandfather in, but that's more difficult to do. And I do have, if anybody's interested, if you go into the American Society of Clinical Pathology, they show you about 15 different ways that you can go if you are, say, an older individual that wants to get into medical technology or medical laboratory scientist, and how you can be grandfathered in for some of your coursework and what other prerequisites you might have to take. Or what your work experience has been. If it's been laboratory based, they will give you some credit for this. But again, it really depends you have to work with the American Society of Clinical Pathology to get them to approve your transcript and your experience. Chris, I don't know what it is for the ASHI program.
The ASHI program, you need the years of experience in a HLA lab first before you can even consider taking it. And then you have to get director's approval. Like the director has to sign you off. And then you actually it's the years that matter the most for ASHI. So the hardest part is getting the experience. So
It is, and that HLA test is hard.
yeah, and there's not a lot of labs out there that are hiring without experience. So I was fortunately enough to get into Gift of Life, which was a general lab that did HLA, which counts as experience. So with that experience on that end, I was able to apply for the exam, take the exam, which is a very hard test in itself. With studying, which there's nothing else out there to study off of, you kind of have to like find your own research. But I think I could piece together, if anyone's interested, I could definitely piece together for you. If you're ever in the HLA field, you can always message me. I'll tell you what to study. It's not too bad, but it's
Likewise, if anybody's interested, I could help them, guide them through the ASCP process. Does anybody else have any other questions? Go ahead.
What do you guys advise for giving tips for those looking for internships after graduation, possibly the biotech lab?
The question was asked after graduation, how do you get an internship into a biotech lab? What are some of the things that can help someone that's graduating get an internship? What and the requirements that might be necessary?
You want me to, I can answer that.
I'm not sure I can answer that.
Yeah, I can answer that question. It's a lot of Googling. So when I was looking for an internship, I was Googling around. I was fortunately enough to have a friend who worked at that lab. So definitely make as many friends as you can, and definitely make as many connections as you can, because some people might tell you some information that's beneficial to you. So fortunately, I had a friend who was actually there as their equipment guy. He was an engineer there and would do all the equipment work on those things. And he had told me about, as I was applying for internships, he was like, yo, you should just try to apply for this one. And I haven't looked back since. So that one was fortunately paid as well. A lot of these internships are not paid. There's a lot of pharmaceutical companies out there that will take you on as an intern. We're talking like Johnson & Johnson, that's up in Princeton. A lot of those other ones that are up there. There's a lot of different companies out there that are looking for interns just over the summertime. Some of them are not paid, but that gets your foot in the door of an opportunity. It's definitely a nice resume builder, which, let's be honest, that's what we want, right? The resume builder to put you out there to make you look better. So whatever you can do to make yourself look better.
The other comment I would have is the National Science Foundation also has internships that they run through a variety of universities. I was fortunate to be able to do one in chemistry, which made me decide I wasn't going to become a chemist.
That's the benefit of some of them. You realize you don't like it at all.
It's really, like Chris says, you really have to expand your contacts, talk to people, talk to your family doctors, talk to, if you're working in a job in a hospital, talk to other people, see what's out there. Just getting an entry-level job sometimes gets you where you want to go.
Exactly.
And if you're applying to medical school, or if you're trying to get into the medical profession, do some shadowing.
We have time for one more question.
Okay, one more question, go ahead.
Based on experience, do you think that to find an internship while you're in school or post-graduation?
The question asked is whether or not it's better to do the internship while you're doing your undergrad or to do it afterwards. I think the answer to that is that it just depends. For example, when I was teaching immunology at Rowan, a couple of my students were doing internships in cell biology in one of the pharmaceutical companies, and that was very, very helpful to them. So sometimes doing an internship during your undergraduate helps you get you focused and motivated and gives you a reason why you're taking a course. think we're good.
Excellent. Well, thank you again, Mr. Gravante and Dr. Paxton, for your excellent presentations today and sharing your career paths with the class. Thank you very much. Thank you. Just wanted to share with everyone that we have, hold on real quick because I need to share my screen. Give me a second. Hold on. There we go. Just wanted to let everyone know that we have 2 more speakers coming up. We have Dr. Beatty and also Dr. Thompson. Also, we have 2 workshops as well. November 4th, we have a cover letter writing workshop. And on November 16th, we also have a personal statements workshop. So keep an eye out for flyers in the hallway, also for your instructors to announce those in class. And thank you again, everyone. Have a good afternoon.
Thank you. Thank you.
Thank you.
Thank you
Take
for
care.
hosting.
Thank you.
Good
Thank
evening.
you.
Bye.